I think our field is changing very quickly. I think as a result of all the MASH clinical trials that we participated in the last decade, we now have 2 FDA-approved medications. I think they are game changers and they change how we approach patients for clinical trials. But also there's room for improvement, and for drug development, we need to improve the efficacy and make sure that we are able to reverse the disease and, most importantly, change its natural history and avoid the development of liver-related complications, and ultimately also make sure that our patients are not dying from liver disease. That would be the ultimate goal for any hepatologist, liver specialist.
Hello, and welcome to Xtalks Spotlight, illuminating insights from subject matter experts and industry thought leaders. I'm Sonia Hunt. In this episode, we're exploring a critical question. What determines success in complex MASH clinical research today?
The clinical landscape for metabolic dysfunction-associated steatohepatitis, or MASH, has been evolving quickly. Advances in patient care, diagnostics, and disease understanding are raising expectations, not just for treatment, but for how research is designed and executed. In this Xtalks Spotlight, we are joined by Dr. Naim Alkhouri, Chief Medical Officer at Summit Clinical Research, to explore what actually determines success in complex MASH research. Dr. Alkhouri shares his perspective on how changes in clinical practice are shaping research expectations, why patient selection plays such a central role, and how teams balance scientific rigor with operational feasibility. This conversation also offers practical insight for sponsors, investigators, and research teams working to design and execute MASH studies in an increasingly complex clinical environment. Thank you for taking the time for this Spotlight interview, Dr. Alkhouri.
Well, thank you for having me. It's an absolute pleasure to be with you.
So to start us off, what do you see as the most important recent advances in the clinical care of patients with MASH? And how are those changes starting to influence expectations for clinical research?
I think we've had major changes in the field of MASLD, steatotic liver disease, and specifically MASH, the aggressive form, because we have now 2 FDA-approved medications. The first one was approved in 2024. This is resmetirom or Resdifra from Madrigal Pharmaceuticals, and more recently in 2025, semaglutide or Wegovy from Novo Nordisk was also FDA-approved. Both of these medications are indicated to treat adults with non-cirrhotic MASH with moderate to advanced fibrosis. And of course, this has affected how we treat patients in our clinics and also how we discuss clinical trials with them. These medications are effective, but definitely there is room to improve efficacy, especially in terms of inducing fibrosis improvement. In addition to the new medications also, we have made significant progress in developing new non-invasive tests that can determine the presence of steatotic liver disease, but more importantly, can determine the severity of the disease, identify patients with MASH and significant fibrosis that will benefit from pharmacologic treatment. And our goal in the next couple of years is to potentially utilize these non-invasive tests as the primary endpoint in clinical trials to replace the need for liver biopsy and histology as the endpoint. So many major changes in 2024 and 2025, and we suspect that 2026 will be another big year for MASH research and clinical care.
So I want to build on that for a moment. From your perspective, where do you see the biggest disconnects between clinical trial protocols and what actually happens in everyday liver clinics?
Unfortunately, there is a big disconnect because in clinical trials, we are not able to include sometimes the sickest patients that may benefit actually from the medications the most. For example, in most MASH clinical trials, we are not able to include patients with uncontrolled type 2 diabetes. Typically, there is an exclusion criteria. If the hemoglobin A1C is above a certain threshold, you cannot include these patients. We do not include patients patients with advanced chronic kidney disease, patients with living with HIV, or patients with type 1 diabetes. And many of these patient populations have high disease burden in terms of prevalence, but also in terms of disease severity. Of course, there's a rationale behind this because we want to make sure that we select patients that are not too advanced, that there is no confounders that may actually affect how they respond to a new investigational product or a new therapeutic agent. So that makes sense to exclude these patients. But unfortunately, in clinical practice, when we encounter these patients, sometimes we don't have the data to support the efficacy and safety of a new medication and these specific populations.
Staying with that idea, patient selection comes up often in MASH research. Why is it such a critical factor when it comes to whether a trial ultimately succeeds or struggles?
That's a very important question. And I think, this has to do with, number one, safety reasons, right? I mean, we want to make sure that we don't include patients that are too sick. They are likely to die from another reason, likely to develop serious adverse events that could be not related to the study drug, but then we have to report everything in the context of a clinical trial. So that's the number one reason is safety. There is also, number two, is efficacy reasons. We want to include patients with significant disease, but we don't want to include those with too advanced disease that maybe it's kind of like the point of no return and the medicine will not be effective in these patients. So we have to be also selective in how far we can push the boundaries and what patients we think we can make an impact for. And maybe some patients, it's too late to get a benefit from a specific medication, and they may benefit from more advanced options like liver transplantation, for example. I think also we need to select the patient phenotype that is most likely to benefit from the drug based on the mechanism of action of the medication. So, for example, if we have a metabolic drug that works on a specific pathway, maybe enriched for patients that carry a higher metabolic disease burden, for example, patients with type 2 diabetes, patients with dyslipidemia, patients with obesity, or maybe a combination of these risk factors to ensure that the medicine has a higher chance of working in this patient population. So several reasons, but we can sum it up as safety reasons, efficacy reasons, and mechanism of action related reasons.
Let's talk about endpoints for a moment. When you're looking at trial endpoints in complex metabolic liver disease, how do you think about balancing scientific rigor with what's realistically feasible to execute?
Our goal is to have this balance between being inclusive and actually having patients in the clinical trial that look like the real-world patient population, so we can have actually the evidence to support treating these patients in the clinic after the clinical trial has ended, but also to keep in mind the specific reasons why we have to be selective, including the safety reasons and efficacy reasons. So I think there is a fine balance here. I think having the expertise in protocol design and working with pharmaceutical companies to help them design inclusive protocols that are also scientifically sound, that will get us to the finish line and help us answer the question if the medicine is actually making an impact in these patients, or if we should stop the drug development and move on to the next asset.
And what role does organizational infrastructure play in supporting consistent trial execution, especially in diseases like MASH, where screen failure rates are high and operations can be quite complex?
I mean, I think this is also a very important question. I'm so glad you asked, but I think this is the most important thing for the success of the clinical trial. When you have experienced sites, especially in the therapeutic area you're working in, so here we're talking about MASH with significant fibrosis or MASH cirrhosis, having a network of sites with experience that can expedite the startup process and make sure that, the sites are up and running in terms of doing the budgets and the contracts contacts and everything it takes to have the study activated at the site. That is key. Having the right patient population, having a database of patients that have been tested for MASH, where you have the disease severity, where you can query the database quickly and identify patients that should be ready to screen for the trial from day one. That will play a significant role. Having experienced principal investigators also that know how to select the right patients to avoid high screen failure rate, but also know how to manage the patients within the trial in case there's adverse events. Sometimes we have also what we call adverse events of special interest. So being familiar with the mechanism of action of the medications, being familiar with the anticipated adverse events, that is also very important. And then retention in these clinical trials is also key because some of these MASH clinical trials can go for 5, 6, sometimes 7 years. You want to keep the patients motivated. You want to get them to the primary endpoint and make sure that you don't end up in a scenario where you lose too many patients along the way, and then you can't really answer in a conclusive way if the medicine made an impact or not because now your sample size is too small. So I think all of these things are important, and you have to think of the clinical trial execution from A to Z. So from answering that feasibility form to see if your site will be a good fit for the trial, to the startup process, to enrolling the patients in a timely manner, managing them in the clinical trial, retaining them in the trial, and getting to the main endpoints.
And to wrap up, what's the key takeaway you would like our audience to remember from this discussion?
Yes, I think our field is changing very quickly. I think as a result of all the MASH clinical trials that we participated in the last decade, we now have 2 FDA-approved medications. I think they are game changers, and they change how we approach patients for clinical trials. But also there's room for improvement, and for drug development, we need to improve the efficacy and make sure that we are able to reverse the disease, and most importantly, change its natural history and avoid the development of liver-related complications, and ultimately also make sure that our patients are not dying from liver disease. That would be the ultimate goal for any hepatologist, liver specialist. I think to do this, we need to continue to innovate and enroll the existing clinical trials and look at new mechanisms of actions. But we have actually several assets in phase 3 clinical trials. Many of these trials are still actively enrolling patients. Many of them are fully enrolled, but they continue to follow these patients for years. So we need to make sure that we retain the patients in the clinical trials. And I think there's no substitute to experienced sites, especially as part of a site network where you can actually enroll many patients, hopefully very quickly in the trial, and manage them properly to help the sponsor get to the finish line and hopefully get more medications that will be FDA approved so we can treat patients in the real world. I think there is room also for many investigator-initiated studies to tackle some of these patient populations that I discussed that sometimes are not included in the main trial. And finally, I think we need to move away from invasive liver biopsies as endpoints, at least for the interim analysis that we do in MASH trials, and hopefully utilize non-invasive tests to help facilitate enrollment quickly and avoid the screen failures related to liver biopsy.
Well, thank you very much, Dr. Alkhouri, for speaking with us today. We really appreciate your time and insights.
Thank you so much. That was a fantastic conversation, and I really enjoyed it. I hope the audience will find this useful.
We look forward to learning how clinical experience, patient selection, and operational realities shape success in MASH research. Thank you all for joining us for this Xtalks Spotlight. We hope you enjoyed the discussion.